Welcome to Jinnious Lab. Today we will cover IB Biology B1.1.1-7 Carbohydrates.
In chapters B1.1.1-7, we would like to learn about carbohydrates. Uh, we can skim through the guiding questions and linking questions, which discusses about what are the functions of carbohydrates and lipids and how they are considered as energy storage compounds, and how the synthesis of these compounds occur with accumulation and become carbon sinks, and also the roles of redox in biological systems. To begin with, we would like to have a basic understanding of what carbon is and how carbon forms four covalent bonds. Carbon is the building block of life, which forms diverse compounds upon which life is used. And this carbon block is used in building carbohydrates, lipids, proteins, and nucleic acids. So basically, lots of the necessary sources and compounds that we require in living. And carbon is, as mentioned before, it's capable of forming four covalent bonds, as shown in Figure 2. 1, 2, 3, 4. And the single bond as a methane CH4 or double bond as in CO2 are examples of carbon forming four bonds with other elements. Covalent bond is a type of bond in which electrons are shared between two neighboring atoms, and what you basically have to know is that it's a strong and stable bond. So there are types of carbon-carbon bonds that carbons form. So the type 1 is a long chain form where the carbon-carbon bonds can form long-chained carbon compounds. And in this case, they do not have any double bonds or triple bonds. They only have single bonds that form a linear structure. And this provides the basic framework for many of the molecules. And this figure involves multiple single-chained carbon-carbon bonds, and this forms the fatty acid structure that displays the long linear form and how it serves as a framework for the fatty acid molecule. Type 2 is the ring, and the carbon-carbon bonds can also be found in chain forms, but also in ring forms. Basically, you can see how this resembles a circular structure. And this is an example of a biomolecule, which is glucose. Carbons can bind to other carbon atoms or non-metallic elements such as phosphorus, nitrogen, oxygen, and hydrogen through single or double bonds. And just for a quick understanding of what a single and double bond is, as you can see, if there's one line, then that's going to be a single bond. If there are two lines between the two elements and when connecting, that is a double bond. In terms of strength, double bond is stronger than single bond. In terms of length, double bond is shorter than single bond. Now, type 3 of the carbon-carbon bond is when carbons bond and form multiple rings. Basically, it's called branched structure. Multiple singular rings can join together to form branch structures of carbon-carbon bond, such as glycogen. Glycogen is a polymer of many glucose molecules joined together. This can be, as you can see, this resembles the branches of the trees, so the structure is called branched. Moving on in B1.1.2-3, we would like to learn about how these carbon bonds or any other monomers are built into compounds via condensation or broken down via hydrolysis. So, prior to learning about these two reactions, we would like to understand what macromolecules are. Macromolecules are types of molecules that are linked by condensation or broken by hydrolysis. Monomers are individual subunits. So basically, it's the most simple form, the basic building block. And you can memorize this by how mono means one. And for polymers, polymers are the ones that are formed by the linkage of monomers. So they're longer and larger. Since it begins with the prefix poly, you can memorize it of how the poly prefix means. Macromolecules are very large molecules that have multiple polymers that are linked together. And you can remember this by how macro prefix means very large. Now, there are four essential macromolecules for humans, which is also hinted in the first section. First, it's carbohydrates, lipids, third, proteins, and last but not least, nucleic acids. Now that's the basics about the macromolecules. And in A1.1.1 to 6, there is a quick brief discussion about the condensation and hydrolysis. It's basically when condensation is forming the macromolecules um, from monomers, and hydrolysis is when molecules or polymers are broken down into monomers. Moving on in B1.1.4 to 7, we would like to learn about sugars. Carbohydrates are composed of carbon, hydrate, which is H2O water. It often exists in the ratio of 1 to 2 to 1 of the carbon to hydrogen to oxygen ratio. Under the carbohydrates there are three smaller levels of carbohydrates. First, it's monosaccharide, second, disaccharide, and third, polysaccharide. You can also memorize this with the prefixes. Mono means one, di means two, and poly means some. Under the category of monosaccharide carbohydrates, there's examples that include glucose, fructose, galactose, disaccharide includes maltose, sucrose, lactose, and polysaccharide includes starch, glycogen, and cellulose. You must memorize this, and you should also know that the disaccharide maltose is formed of glucose and glucose, that sucrose is formed of glucose and fructose, lactose is formed of glucose and galactose. This is a tip to memorize the monosaccharides that make this disaccharide, which is G plus G, glucose plus glucose is maltose, glucose plus fructose is sucrose, and glucose plus galactose is lactose. Now, to learn more about these polysaccharides, disaccharides, and monosaccharides, let's move on to this chart where you have to memorize everything on this chart. So, polysaccharides examples include cellulose, starch, and glycogen as mentioned. And cellulose function is to provide structural support in the plant cell walls and porous that allows the water to flow through it. In terms of its structure, it is built of beta D glucose, which is joined by a 1,4 linkage. It is unbranched and it has a linear form. The chains are cross-linked by hydrogen bonding, and as we know, hydrogen bonding is a very strong bond. Now, for starch, the function is for plant energy storage, and the structure is unlike cellulose, alpha glucose, which is also joined by 1,4 linkage. It has two forms, which is amylose and amylopectin. Amylose is unbranched 1,4 glycosidic linkage, while amylopectin is slightly branched because it has both 1,4 and 1,6 glycosidic linkage. It has more efficient energy storage because both bonds are present. And amylose and amylopectin are both coiled and insoluble. For glycogen, its function is to store energy for animals. The glycogens are always stored in liver and skeletal muscle cells. It forms tiny granules in cytoplasm, and the structure is alpha glucose, which is joined by 1,4 and 1,6 links. It is insoluble. Now, to learn about the glycosidic linkage, which is mentioned in the formation structure of the polysaccharides, we need to understand the isomers and the glycosidic linkage of glucose. So there are two isomers, main isomers in glucose, which is alpha and beta glucose. For alpha glucose, the OH, which is a hydroxyl group, exists below the carbon 1. As you can see, the hydrogen is facing upwards, while the hydroxyl group faces downwards. For the beta glucose, it's the opposite. The hydroxyl group faces upwards, while the hydrogen faces downwards. So, due to this isomerism, the linkage can exist in both 1,4 or 1,6 form. As you can see in this diagram, the 1,4 linkages is when the uh, linkage occurs between carbon 1 and carbon 4. And the 1,6 linkage occurs when the bonding is between carbon 1 and carbon 6. So these two are the glycosidic linkage types which occur due to the different isomers of glucose. Moving on to the functions of carbohydrates. Energy is one of the functions of carbohydrates. It is oxidized to produce biological energy ATP via cellular respiration. So basically, carbohydrates are a form of energy that can be converted into biological energy for cellular respiration and use in body. It is also soluble, and the solubility serves as its function. Glucose is a polar molecule due to the OH groups that are attached to the carbon ring, and this allows it to easily dissolve in water, and this allows the glucose to be easily delivered in the blood for cellular respiration. For structural support, for example, cellulose. So carbohydrate type included cellulose in the polysaccharide, and this provides structural support for plant cell wall. Cellulose is a polysaccharide composed of beta-glucose, which serves as a structural support of plant cell wall. One cellular chain, the beta-glucose alternate in its orientation to form a straight linear cellulose chain. As you can see in this diagram, it forms a straight chain structure, and uh, because the multiple cellulose chain interaction is conducted with hydrogen bonding, it is a very stable and strong bond. Cellulose is very strong due to the number of hydrogen bonds that are cross-linking between the strands that forms the microfibrils. These are possible due to the beta-glucose alternating in its positions from a straight linear cellulose 1,4 linkage. And the significance is that the hydrogen bonding between cellulose strands increase the plant's structural integrity and mechanical stability. Without cellulose, cells wouldn't be able to withstand the forces of osmosis and would collapse under their own weight. Another function of carbohydrate is cell-to-cell recognition. If you look closely to the phospholipid bilayer, you would also see the glycoproteins that are attached to the bilayers. And glycoproteins are basically proteins that which are condensed with carbohydrate. So it's a compound where the carbohydrate and proteins are attached together via glycylation. And this can perform cell-to-cell recognition, which the immune system allows which allows the immune system to recognize the cell as a cell which is belonging to our body. The example is the ABO blood type decision by the glycoproteins on human red blood cells. As you can see in this image, the A has the glycoproteins that act as a flag in our body to inform our immune system that this is the A red blood cell. And for this B, for AB, both A and B, and for O, there are no glycoprotein signals. This is also the reason why the blood transfusions are not compatible with different blood types, and also the reason why the blood type O can share the blood to AB and AB, all of them, because they do not have glycoproteins. However, for O, they only can accept O because they can only accept the red blood cells without any glycoproteins. This is a quick diagram about compatibility depending on the ABO blood types here. And here is an appendix of how to memorize the cellulose, starch, and glycogen's source, subunit, bonds, branches, and the diagram, along with its branched, linear, or coiled shape.
For IB45 Jinnious Lab, don't forget to subscribe and like.